Frank and John for bringing us through the last 20 years since microRNA was first discovered by Frank. As the previous three speakers alluded to, I think there has been a lot of excitement about the biology and the potential of microRNA in illustrating some of the information that's very unique to microRNA that's not found in DNA, mRNA, or proteins. But there has also been equal number of disappointments during this whole process in the last 20 years. What we're very glad is the biology of microRNA is becoming very clear and its potential in commercial application. And what we are also glad is the challenges seems to be in the process of how we are discovering these biomarkers and developing the assay. My belief is that all process challenges can be resolved, and this is why we're very glad that the Singapore National Standard SS656 is now in place to guide all of us, not only in Asia, but I think across the globe, everybody that's working on microRNAs, to then have a unified approach to bring a biomarker from discovery all the way down to a clinically implementable assay. So I'm going to take the journey of developing GastroClear, which is the world's first stomach cancer microRNA screening test done in blood, and share with you what are the many challenges we have learned, and also I think some of the valuable lessons that we believe will benefit everyone else. So this test is designed, developed, and manufactured in Singapore, and it received a home country approval in May 2019. And this is the first stomach cancer microRNA blood test that's been approved globally. Why did we look at stomach cancer? Well, it remains to be the third most deadly cancer in the world, although the incidence rate has been dropping. But basically, by the time stomach cancer patients show symptoms, it's often very late. So screening measures are extremely important. Across the globe, Japan and Korea have the highest prognosis, the best prognosis for gastric cancer patients, because they implement national endoscope-based screening, which is frankly is not applicable in many of the other countries without the level of infrastructure, and also have medium to low prevalence of gastric cancer. So when we first got into developing a stomach cancer screening assay, it was really out of the request of many clinicians to ask for a tool, a simple tool, which is accurate, affordable, actionable, to help screen the asymptomatic population before subjecting or referring the high-risk patients to do an endoscope. So its clinical utility is in looking for early detection in asymptomatic patients, and at the same time help to make the whole screening process more affordable for the entire healthcare system. It was an 8-year effort starting in 2012, led by Professor Yeo Kei Guan, our lead PI from the Singapore Gastric Cancer Consortium, but also combining the effort across the globe. In Singapore, we have NUH, TTSH, and John has mentioned DXD Hub at its establishment in 2014 really helped guide us through the whole process of developing and designing the assay. School of Public Health in NUS helped us to look at the health economics, and of course the foreign hospitals that has participated in the whole effort in Korea, in Japan, in China, as well as Vanderbilt in the United States. So this was really a collective international effort in the last 8 years that brought the test to its fruition. Now, this is a PCR-based test, and I think what COVID pandemic has brought us is really globally everyone now can talk about PCR. Everyone seems to know about RNA, which is great for molecular diagnostics. But very briefly, which I believe Ling will share in a lot more detail, the clinical laboratory workflow of this is a 4-step process where the microRNA is isolated from the clinical sample, in this case serum, converted into a cDNA, detected using a qPCR process, and subjected to data analysis, where the sample to result time within the lab can be done within 4 hours. And as Yulia has alluded to, there are many analytical and biological variants that can happen across a whole process. So how do we best control that so that we can get the best biology out of these microRNAs? We're also very glad upon approval of GastroClear in Singapore. This is the only microRNA test that was featured in the Nature Biotech article when we talk more about cancer liquid biopsy, early detection, and precision medicine, alongside with the DNA sequencing tests, methylation tests, and some of the earlier protein tests. So officially, it was a 6-year journey from a biomarker discovery to the finishing of the clinical validation prior to a regulatory submission. And why did we take 6 years? And that was precisely the many challenges that Zhou Lihan has highlighted. In order to fully illustrate the potential of these biomarkers, it took us many iterations from discovery to verification to assay development and ultimately the clinical validation. And the prospective clinical study alone took 3 years, from 2016 to 2018. Why did we take that long is because we wanted to illustrate really not only the clinical performance, but the clinical utility of a microRNA test. So we enrolled on a high-risk population basis, a total of 5,282 participants, all of which had a blood test, and all of whom had an endoscope as a reference point. So within this population, there was 2.5% of cancer, and the blood was collected prior to any of these diagnoses. So, and after going through this whole process, eventually 4,566 of these were included in the analysis. We had 115 of the gastric cancer cases, 10 high-grade dysplasia, and what the microRNA test was able to show is gastric cancer is one of the diseases where we really lack a good blood-based biomarker. And what has been implemented in high-risk countries in Asia, China, Korea, Japan, you will see H. pylori, which is a bacteria that causes infection in the stomach, which puts the patient at very high risk of developing gastric cancer, pepsinogen 1 and 2. And when we compare the microRNA assay in the prospective clinical validation, we were able to elevate the accuracy as compared to the existing biomarker by more than 20% in the AUC. And even in high-grade dysplasia, which is known as the stage 0 gastric cancer, the AUC is also 25% better as compared to existing biomarkers. So we are very excited about the performance of these biomarkers. And what's even more exciting to the clinicians is the great correlation of the sensitivity of these assays in detecting early stage of gastric cancer. And if you look at figure B over here, there is also a good correlation according to the lesion size. So this is something that John and Frank has alluded to. Many of these microRNAs come from the cancer tissue itself, and of course it makes sense that when we have a bigger tumor, there are greater shedding of these microRNAs into the circulation, and therefore a higher sensitivity. And upon approval last year in May, what was I think all of us got very excited about is we managed to implement this in one of the community screening projects launched by the Singapore Prime Minister. We offered the test to screen 242 elderly, asymptomatic individuals, and we're monitoring 2 of those that has been identified as high risk. So coming back to the standard, why are we so excited? Is because we have learned so much. We have made so many mistakes along the 6-year journey, and we wanted to make sure other people don't repeat the same mistake, and we wanted to make sure our regulators understand the pros and cons of developing microRNA diagnostics and what to watch out for. So John has also shared part of this, and Zhou Lihan has alluded to this as well. It is a very long process, and each of these steps must be aligned and controlled very well, or else any failure in any one of these steps could result in the eventual failure of the test, which I believe is why up to today, not as many RNA-based tests has been implemented. So why is microRNA unique? There has been other standards that talk about the development of DNA tests, the development of protein tests, and call to that is really the size of the microRNA. It's extremely small, so the typical chemistry that works for the bigger analytes really do not work for microRNA. and the considerations in analytical and clinical variability would be different. So you look at this graph. In order for something to be able to implement clinically, the assay must be robust itself. The left-hand side is a typical research grade assay that sometimes works, sometimes doesn't work very well. But in order for clinical implementation, we really need something as robust as what's shown on the right-hand side, and that has to be consistent across the hundreds and thousands of microRNA, and that is not an easy task. And what makes this even more challenging is when we look at the circulating microRNAs, the very high abundance ones, which Yulian mentioned, that's been shedding by various tissues, those are not as informative as some of the lower expression ones, which are secreted only by selected cells or tissues. And coupling the small size of these microRNAs and the less abundance of these microRNAs, the development process, the assay, becomes extremely important. So we became very obsessed with quality, with control, and this is really, I think, the slide that we use convince Frank and Yulian to work with us, where we talk about excessive control of every single possible step in order to remove the analytical variability to fully illustrate the biology of microRNA. And again, an often forgotten part of the quality control process is sample collection, which many of our researchers or even commercial companies may or may not have fully understood the importance of quality control in the sample collection process, because ultimately for any test, it's garbage in, garbage out. So we are very glad in the SS656, we don't only talk about the control of the development process, the thinking, but also we highlighted the need to control for sample collection and processing. So I won't go into details, but if you look at the way the SS656 is structured, we believe this will offer really a great framework for anyone who's interested in microRNA biomarker or candidate discovery to think about what are the processes, what are the pros and cons, and what are the things to watch out for. And we're hoping that this will also provide a framework to the regulators, not only in Asia, and also in FDA, that this is how we are approaching development of microRNA, which is different from the other assay development. And hopefully, by the collective effort, we'll be able to push more microRNA IVD tests through the regulatory approval and a greater clinical adoption. So we ourselves are very excited to apply SS656 to guide any of our other microRNA assay development, as well as the broader sense of RNA diagnostic tests. And the first one we are applying that to is the lung cancer microRNA test, which we are conducting a fairly large global effort. And the reason, and it's also mentioned in SS656, is the key to any RNA or microRNA-based expression level assays is to make sure that the performance is consistent across different countries, with different ethnic group, with different disease biology. So we are applying that to the lung cancer test development. And interestingly, as it was mentioned, this year we pivoted some of our capabilities to assist in the national effort in ramping up the COVID-19 RT-PCR test. And interestingly, we actually applied many of the learnings that's written in SS656 to the development, manufacturing, and validation of Fortitude, which is essentially an RNA-based test. So glad to share that the blue dots are all the countries that Fortitude has been deployed to. And we look forward to the day when microRNA tests will be deployed in as many countries as the COVID-19 test. So with that, I just want to say our gratitude as an industry member to the national body and the many people who has participated and launched the SS656 in really a record speed from November to now. And we also like to thank our resource members, Frank and Yulian, for guiding us through this whole process. So thank you very much.